Good afternoon. Any questions before we get started today? All right, let's go ahead and talk game plan then. Today we are going to finish off our immune response. We've talked about our first two lines of defense. Now we're going to talk about our adaptive defense, our third line of defense. If there's time, we'll start talking a little bit about the endocrine system. I strongly encourage you to start working on that this weekend. It can be a bit challenging. So if nothing else, we'll talk about the handout that you have, the study guide for that to help you to get started on that. 3 more assignments to do in this section. Your PhysioX exercise 12, activity 3, just activity 3, that is due on Tuesday. On Thursday, your Labster about the immunology, introduction to immunology is due. And then obviously your group presentations. And again, remember for those group presentations, you are turning in your outline. Again, you can turn it in as soon as it is done, but it has to be turned in by 5 p.m. on Monday. I want it combined into one PDF. So even if it's a 3-page outline, I don't want it as 3 separate PDFs. I want it combined into one PDF to make it easy for everybody. Make sure your name is on it. Make sure the name of your disorder is on the handout. And again, it is important that we get this available for students before the presentation. So if you do not turn it on time or you forget to submit it, it is going to be 10 points off of that 50-point assignment. That is a huge chunk. And so again, that is a 20-point penalty, a 20% penalty. So don't make that mistake. It is very important to do that so that everybody's prepared for Tuesday morning when we start our group presentations. And then our lab and lecture exam is on Thursday. All right. Questions on any of that? Chat question. No, so yes, it's one outline per group. So the entire group should combine their information into one handout. So I want one outline, because again, it's a group presentation. You're not doing 3 individual presentations. You're doing one presentation. So I want one outline for the entire presentation with everybody in the name, everybody in your group's name on it. All right. Any other questions? All righty then, let's get started. We are on to our third line of defense, right? Our specific or adaptive immunity. And again, what makes it specific is that it is specific per antigens. So we talked about that makes it very versatile. You have a whole key chain worth of keys that can fit in locks to be able to be efficient in identifying and destroying harmful pathogens. And again, where does that versatility come from? Where is the versatility? Where is the, which shape keys you have on your keychain determined? I can wait. Still waiting. Hazard a guess. You guys can't hear me, right? Here, hold on. Oh, I don't have my microphone point. I know I have a microphone. This microphone's better. Can you hear me better now?
Yes.
Excellent. All right. So again, your specific defense has a starting point where, you know, the versatility of your immune response, what shape antigens you have to be able to identify foreign things. Where does that come from? Where does that process start? There you go. Genetics. Absolutely. It starts with your genes. Absolutely. All right. And again, the advantage of this specific defense is that it is very effective. It is systemic throughout your entire body. And the most powerful thing about it is that it has that memory. Right. But as we talked about, if there is a downside, what is the downside?
It's slow.
Time and symptoms.
Yeah, it's slow to build up. It takes time for it to build up the first time. And so typically you get symptoms from the illness while you're building up defense. And, you know, theoretically, if the illness is severe enough, it can damage you or even kill you before you get a chance to build up that defense. Right. And again, one of the things that we talked about is we're talking about these systems independently, but remember, they really don't work independently. They all work together to provide that defense at the same time, and they often help each other and enhance each other. All right, again, this specific defense is because of antigens. This is a concept that we have already talked about. We talked about it starting back when we were talking about blood types. Antigens are the tags that are on the surface of our cells. They are mostly proteins, but they can be combinations of proteins and lipids and carbohydrates. But what we haven't talked about is these antigens in as much detail. Typically, antigens can be complete or incomplete antigens. To be a complete antigen, there are two main requirements that the antigen must have. The first is it must have what we call immunogenicity. Immunogenicity basically means that it can stimulate a lymphocyte to divide. We have lymphocytes, and we'll talk about the anatomy of our lymphocytes. And one of the things we'll learn is when we stimulate. Great question, Susan. Yes, I would say that yes, if you had ways to buy time for your body in some way to build up a defense, it would, for that time, absolutely, right? So I think a great example of it is that snake tamer we talked about, right? You get bit by that snake and you have to take the anti-snake venom, and then the next day you have to do the same thing, and the next week you have to do the same thing. After being bitten 20 and 30 times and having to take that anti-snake venom all during that time, do you think after 30 exposures to that, that person's body might slowly start building up a little bit of a defense to that snake venom? Right, so by taking all of those anti-venoms, they've slowly been able to build it up and become a little less, a little more immune to it, a little bit more resistant to it. Sure, absolutely, something like that could theoretically happen. All right, one of the powers of our specific defense is, unlike our natural killer cells, where you have that one lone night watchman running around the house by himself, when a lymphocyte gets activated, the first thing it does is divide rapidly to produce many new cells to give us a very large response. So the key to this is that there is, and we'll talk about what that tag is, there is a structure or a receptor on the surface of our T cell that allows it to bind to our pathogen, right? The antigen of that pathogen. So here's my antigen, here is it, my pathogen, here is its antigen, and let's pretend that that lined up. It's able to bind to it and stimulate that division. That ability to stimulate the division of this activated cell that's a little too big. Of this activated cell is what basically we mean by saying immunogenicity. So the specific cell that is specific to this antigen, that lymphocyte is able to divide in response to that exposure. All right, and that is an important part of being an antigen. However, the first part of being an antigen is that reactivity, that ability to activate the lymphocyte, to activate those antibodies. So when it comes across it the first time, it actually has to activate the lymphocyte so that it can then be immunogenic. The reason for this is that there are some incomplete antigens. An incomplete antigen is a small molecule, most small molecules for that matter, are not immunogenic. However, some of them can be reactive. And that's the problem. The problem is that they activate these immune cells inappropriately. When this occurs, we call them, as we mentioned, an incomplete antigen, or we call this a hapten, right? What this means is that when some harmless piece of protein typically binds to another protein in the body, or a cell in the body, or something along those lines, it is able to cause that reaction to our immune cells and stimulate and activate our immune cells, right? Often, what do we call that when we get this inappropriate activation of the immune response?
Allergy.
Say again?
Allergy.
Autoimmune.
Allergies, exactly, right? It triggers that inappropriate response. We call it allergies, or more specifically, we call it a hypersensitivity, right? Some of these are relatively benign. For instance, the pollens that are released from the grass by cutting the grass, or the pollen, or not the pollen, or the dander that is released from a dog inside of a house, or the dried saliva of a cat, right, that gets into the circulatory system of a house. These are the types of things that are not harmful, right? They're not immunogenic. They're not the type of thing that by themselves can trigger an immune response. But when bound to the cells of our body, like in our mucous membranes, they can trigger a reaction. And that leads to the inflammation, that leads to the runny nose and the watery eyes, right? And while that is bothersome, right, there are other hypersensitivities, things like bee stings, things like nuts, where as a result of those, not only are you just stimulating the mucous membrane, because those still get affected, your mucous membrane still thicken and produce mucus as a result of that, but also as a result of that, you get a rapid dilation of the blood vessels systemically. And what did we call that situation again? That type of reaction? Things like
Anaphylaxis.
Yeah, anaphylaxis, absolutely. An anaphylactic shock occurs because of those hypersensitivities or those allergies. So while often they can be relatively benign, they can also be life-threatening as well in that. All right? Questions on that? All right, so we left off last class and we were talking about all the different types of immunity. And what type of immunity did we say was actually our immune response?
Naturally acquired.
Okay, you get partial credit. Naturally acquired.
Active immunity.
There we go, excellent. It is our naturally acquired active immunity, absolutely. We are exposed to a pathogen, we do the work, we get the memory, all right? For this immune response, we basically have two related processes. They are what we call our humoral immunity, right? Humoral refers to what again?
Blood and lymph.
Sure, absolutely. They are the fluids of the body.
Humors of the body.
Right, the humors of the body, the fluids of the body. And you're absolutely correct. The primary ones of that are lymph and blood plasma. Of course, if you think about it, if it is in the blood plasma, then that also means we're also talking about interstitial fluid as well, because those intermix with each other. Oops, I guess if I spell lymph correctly. Right, so it is those fluids of the body that we are talking about. And humoral immunity is also what we call antibody-mediated immunity, because this is the process where we are producing antibodies. Where's my... Here. The advantage of this humoral immunity, or this antibody-mediated immunity, is that it attacks the pathogens directly. Possibly even before they enter the cell. This is different from cellular immunity. Cellular immunity is where we have cells of our body that attack other cells of our body. Right, so in this case, the cellular immunity is where we are directly attacking either infected or in some other way abnormal or foreign cells. Oops. Right. This means not just cells that are infected by a virus or a pathogen, but these are parasites that we could have. These are cancerous cells. And unfortunately, as we also talked about, these could be cells that have been purposely put in our body. Transplanted cells, like a transplanted liver or a transplanted kidney or a transplanted lung. Things along those lines. All right, so we have these two different types of defenses that are related to each other, but obviously have different targets and therefore different functions. And we have cells that help us in this process. The primary cell of our immune response, say again, so in humoral immunity, the pathogens aren't attached to any cells. Typically, well, let's say it this way, it can. Is it possible to also attack a pathogen that has affected a cell? Yes, but typically the goal is of the humoral immunity, is, since it's antibody related, is to be looking for that antigen on the specific pathogen itself and to try to disable that pathogen before it has a chance to attack the cell. So yes, typically before it is attached to the cell. We get to directly attack the pathogen first. Well, not first, but with that humoral immunity. All right. Great question. Any others? Can we have the writing again, please? Sure. That? Oh, you mean what I wrote? That? All right. Ready? All right, stun silence says yes. Excellent. All right, cells of the immune response. Primary cell of the immune response are lymphocytes, right? As we talked about, they're the ones that are responsible for our immune response. Where do lymphocytes originate again? Red bone marrow. What is the primary stem cell of a lymphocyte? Primary stem cell, hemocytoblast. What is the secondary stem cell of a lymphocyte?
Myeloid.
Myeloid?
No,
Lymphoid.
Lymphoid. Lymphoid stem cells. Excellent. See, there you go. Some information we remember from the first section. And these cells that are produced from these lymphoid stem cells, remember lymphoid stem cells are a unipotent stem cell, then become what we call immature lymphocytes. These immature lymphocytes then migrate to one of two locations. And what are the two locations that they can go to mature?
thymus
Or
red bone marrow.
yeah, red bone marrow. Excellent. So they are either going to stay in the red bone marrow or migrate to a different region of the red bone marrow, or they're going to migrate to the thymus. Those that stay or find different bone matter to mature in, B for bone, become our B cells. And of course, those that go to the thymus become what?
T cells.
B cells, exactly. It is the B cells that are responsible for our humoral immunity. And they are the ones that end up producing antibodies. Where the T cells that develop in the thymus are the ones that are responsible for our cellular immunity. And they are the ones responsible for that hot cell-on-cell action, attacking foreign, infected, and abnormal cells in the body. All right. We have a pretty picture that goes along with this, but we have to talk a little bit about vocabulary, right? When I say immature, I don't mean immunocompetent. All right, well, let's say it this way. What do I mean when I say immature?
Not mature yet.
But what does that mean? What does it mean to not be mature for these particular cells?
It hasn't been taught self versus non-self.
Okay, that's part of it. It hasn't been taught self versus non-self yet. It hasn't been activated. That's true as well. But there's something even more basic than that. if you were to take a virus, the right virus, and rub it all up and down on that immature lymphocyte, what's going to happen?
Nothing.
Nothing, exactly. Because immature cells are not capable of immune responses. So if the cell, even if it is the right pathogen, if the cell is immature, it is not capable of an immune response. Whereas a cell that is immunocompetent or is mature is capable of an immune response. Now, just because a cell is capable of an immune response, does that mean that it has been activated? Does that mean that it has come in contact with its pathogen?
Not yet.
No.
Not necessarily. No, not yet. Exactly. In fact, like we talked about, if you've never been to South America, if you've never been to Panama, you may have cells in your body. You may have mature cells in your body that can protect you from malaria. But if you've never come in contact with malaria, then those cells are never going to be activated. And a cell that has not been activated, we call naive. Right? So there's a difference between being immature and being naive. A naive cell is an immunocompetent cell. It's a mature cell that has never been in contact with its pathogen. And you will live your entire life and die with some of your immune cells, hopefully, with some of your immune cells that stay naive. Hopefully, you will not come in contact with every disorder that your body is there ready to protect you from. I wouldn't say prematurely activated. I wouldn't use that term. I would say inappropriately activated, and that typically when a immunocompetent cell is inappropriately activated, that typically is due to some type of hypersensitivity or some autoimmune disorder or something along those lines. Great question. I would say, sure, for our purposes, let's say that they're equally distributed between the thymus and the red bone marrow. We probably have, although we know that that's not entirely the case. Yeah, let's say for our purposes, sky is blue, yes, equal amounts of both. All right. Questions on this? All right, now we know the difference between immature and naive. Now we understand the difference between mature and immature. Let's talk about how we mature these lymphocytes. Now, the study of the lymphocytes has primarily been studied in T cells. But everything that we understand is that it's the exact same process that occurs in our B cells as well. So we will be talking about our T cells primarily, but our understanding is this works the exact same way in our B cells. This process requires about 2 to 3 days, and basically what happens is our lymphocytes have to pass 2 important tests. The first of these tests is the process called a positive selection. All right. If you remember in the last class, and I'm going to cheat and I'm going to go to our whiteboard for this for a minute. We will talk about this in more detail in a moment, but for right now, let's draw it this way. This right here is a cell of our body. And as we talked about in the previous class, our body cells have proteins for self and non-self. Well, for self, I should say, right? They have proteins for us that we've been drawing all these different shapes, circles, squares, triangles, that we use to recognize things that are us. And if you remember, one of the things that we talked about was a major histocompatibility complex. And the good news is once you write that out once, you can abbreviate it MHC. Now, as we will learn, there are actually two classes of those, but we're not going to worry about the different classes just yet. We will get to that. But the point is, every cell in our body has these major histocompatibility complexes. And these are the proteins, these are the antigens that say, this is me. Right? This is basically what we call the self-antigen. And so if our T cells are going to be able to produce an immune response, one of the things they have to be able to do is they have to be able to bind to that major histocompatibility complex. They have to have the right type of receptor to bind to it. If they don't, right, if their receptor is somewhat wonky shaped, where it can't form that bond with the major histocompatibility complex, then this T cell is not going to be able to be activated. And if this cell cannot be activated, is it going to do us any good?
No.
No. And if it's not going to do us any good, do we want to keep it?
No.
No. So if it can't do us any good, then basically what happens in this cell is we are going to trigger apoptosis. All right, you're no good to me. Time for you to die. And we will kill this cell that is unable to bind to that self-antigen. All right, so far so good?
Yes.
This is our positive selection. So must be able to recognize that special self-protein on the cells called the major histocompatibility complex. All right, you with me so far? It only gets worse from here. So if this doesn't make sense, let me know now.
Why it's positive, not negative?
Because in this case, we want it to recognize this particular protein. So yes, it recognizes this protein, or no, it doesn't. If it recognizes it, then that is a positive, and that's a good thing. So this is our positive selection.
Okay.
Right? The goal, the correct answer is yes. I recognize the major histocompatibility complex, and that is the positive selection. All right? So again, it gets worse from here. So any questions on this?
Recognize means I recognize it's not me, or I recognize it's me.
I recognize that it is me. Right? This is the self part. We are recognizing self. It's a positive recognition of self.
So not the next one that cannot be activated because it's not me. It's not about this case.
Right. So in this case, right, so the cell A here is able to recognize that major histocompatibility complex, and cell B cannot. So since A can recognize it, A positively recognizes that self-protein. That is a positive selection. That is a good thing. Cell B cannot bind and recognize to the major histocompatibility complex. And so that means that it's not going to be able to be activated. And if we can't activate it, it's not going to be any good to us, and so we need to get rid of it.
So in this case, it's negative?
No, it didn't pass the positive selection.
Okay.
Okay? So this one passed the positive selection, right? This one passed the positive selection, and this one failed the positive selection. All right?
Yes.
I don't mind these questions because, like I said, it just gets worse from here. So I need to make sure you guys understand this before we go to the next step. All right, excellent. Stunned silences mean that I understand everything, make the test harder. Perfect. All right, then let's switch to our negative selection. For that, I need to do some cheating. I will get rid of that and that and that and that and that and that and that. All right. Now, let us talk about what our major histocompatibility complexes do. What our major histocompatibility complexes do is they are actually proteins that are produced inside the cell. And when these are produced inside the cell, so let's go ahead and draw one of these bad boys inside the cell. Essentially what they do, as we know, cells are protein factories, making massive amounts of proteins. So we're constantly getting sequences of amino acids and taking those subunits and putting them together. And we know the Golgi apparatus is cutting pieces off, and there's constant protein synthesis going on in this cell. Well, it turns out what the major histocompatibility complex will do is it will grab some random piece of a protein inside of the cell and hold on to it, so that when it then expresses itself on the surface, it holds that little piece of one of our proteins. This is a self-antigen, and it will hold that self-antigen on the surface of the cell. So in this case, this is a small piece of one of our proteins. And it is a self-antigen, all right? It's a piece of protein from one of our cells. These are things that our cells are making that we want our cells to use and can be used by the cell. Now, do we want our T cell to be able to recognize and be activated by a self-antigen? No. So in this case, what we have is our negative selection. Right? In this case, cell A happens to have the binding site that would allow it to match up with, I got to cheat a little bit, move this. So you notice this binding site would let it match up. Let's pretend it matches up and binds to that there. That means that this cell could be activated by one of our proteins. Would we want that? Would we want our T cells to be activated by our proteins?
No.
No. So this failed the negative selection. Cell B here on the other side has a binding site with a different shape for a different antigen. Is that going to bind to our antigen and recognize our antigen as being foreign and want to be stimulated and want to be reactive to that?
Yes.
That's going to be able to bind to this antigen?
Oh, no, it cannot.
No, it's not. So notice in this case, it passed the negative selection. It is not reacting to any of our proteins. So we need a T cell that can do two things. And again, I say T cells, but I also mean B cells. To do two things. They must be able to bind to major histocompatibility complexes and not react to our proteins. And as long as it can do those two things, then it can be part of our immune response. So this is our negative selection. With this negative selection, the T cell must not be able to recognize any of the self-antigens. And again, I want to make sure that we mention this here. These self-antigens, remember, are being expressed by the major histocompatibility complexes. So they cannot respond to any of the antigens, the self-antigens that are being expressed by the major histocompatibility complex. And we say these cells are self-tolerant. meaning that they're not activated by our proteins. Now, what percentage of these T cells do you think meet both criteria? Yeah, exactly. Only about 2% of the T cells survive this process. Because again, let's go back to the picture as is being asked. Remember, if you fail the test, again, we need to trigger apoptosis in this cell. We need to kill this cell, because if we don't kill this cell can kill us. All right. Questions on that? All right, excellent. Stunned silence means we understand. Let's move forward. So let's do that. Lymphocytes are a very important cell of our immune response. Oh, and here's the pretty picture. Shows that where, again, we see starting in the bone marrow and then either maturing in the thymus of the bone marrow. Once they're mature, once they're immunocompetent, they go and hang out places like the lymph node, the spleen, other lymphatic tissues. Small percent, 2%, seem to be circulating around. I don't think of it as being inefficient. I think of it as being overly cautious. Right? These things don't necessarily want to be rushed. Right? Do we want to have lymphocytes that may or may not react to our body? It's okay, as long as it only kills 200,000 of our cells. That's virtually nothing, right? Isn't that what we've learned recently? 200,000 is virtually nothing, virtually no one. Right? So who cares if it's not super efficient, right? There's collateral damage. That doesn't matter. Yeah, these are cells that could potentially kill us. We want to be very careful and very cautious. All right, and that's what we have. These are deadly weapons. And so we want to make sure that we are very, very careful and very cautious in what we allow to float around inside of our body. I mean, isn't that the problem with all the, many of you are doing studies on autoimmune disorders where our immune system attacks our own body. And anybody who's studying an autoimmune disorder, do any of them sound particularly fun? I'll take the sun silence as a no. No, typically not, right? So again, so yeah, we want to be really careful about which ones we activate. All right. All right, so like I said, lymphocytes are one of the important cells of our immune response, but they are not our only cells. I'll go ahead and mute that, and then they can take care of that. Excellent, but also our phagocytes as well. Which cells did we say primarily are the ones that are going to be activated in our immune response?
Neutrophils.
Neutrophils and
Monocytes.
there you go, and our monocytes. Excellent. Those are our phagocytes, and we talked about mobilization of those, and we talked about phagocytosis. We've talked about those processes. We also started talking about, and we're going to continue to talk about, antigen-presenting cells. As we've talked about, cells like phagocytes take a little piece of the virus that it's broken down, or the pathogen that's broken down, and expresses it on the surface of its plasma membrane. And guess what protein it uses to express that antigen on its surface? There you go, absolutely. It uses the major histocompatibility complex. Oops. To express it on its surface. Obviously, as we talked about, phagocytes are antigen-presenting cells, but there are others. Dendritic cells. I think we talked about these last time, but remind me again, where do we find dendritic cells?
Skin.
Skin, there you go, excellent. Infected cells, right? Infected cells have major histocompatibility complexes, but they are expressing, right, viral proteins from inside of them, different proteins, right? Because remember, the virus hijacks the protein process system. Our Kupffer cells in our liver are phagocytes that are very important in stimulating our immune response. Remember all the stuff that we talked about, all the nutrients and all the materials we absorb from our digestive system go to the liver first before it goes anywhere else in the body to be processed. So these Kupffer cells are important guards, guarding the intake of materials from the digestive system into our body. And as we'll learn, our B cells actually also are considered antigen-presenting cells, and that is an important role in how they are activated. So we'll talk about that when we actually talk about B cell activation. All righty, those are the players. Let's talk about the process. And we are going to start with cellular immunity first, because typically cellular immunity occurs first. So we will talk about our cellular immunity. Our cellular immunity, of course, uses the T cells, as we mentioned, whereas it is the B cells that are responsible for our humoral immunity. And T cells come in different flavors. There are primarily 4 main types of activated T cells. All right, again, T cells are the ones that are responsible for our cell-mediated immunity. They directly attack, and they're the only cells that in our immune response that directly attack other cells. And there are 4 main types. There are helper T cells. What do you think helper T cells do? They help, exactly.
T cells?
They help to stimulate our immune response. Cytotoxic T cells, which are sometimes affectionately known as killer T cells. They're the ones that are actually doing that cell-on-cell action. Memory T cells, and our 4th type, which are regulatory, or sometimes also referred to as suppressor T cells. So there are 4 main types of T cells, and we need to talk about all 4 of them. Now, again, we're doing this first because, as I mentioned, T cells are typically activated before the B cells are. But remember, as we talked about, they are responsible for attacking cells. And so they're typically not activated by the pathogen directly. Instead, they need the antigen from that pathogen expressed to them by an antigen-presenting cell. That's why these antigen-presenting cells are so important. It's these antigen-presenting cells that are going to stimulate, or you could say activate, the T cells. All right?
Those that from phagocytosis, right?
Right, so definitely, so remember, phagocytes are one example of that. But remember, we also said abnormal, foreign, and infected cells can also. So really, there are 2 main types of antigen-presenting cells. Our phagocytes, and phagocytes would also include things like the dendritic cells and the Kupffer cells. Oops. In the liver. So there's really 2 main types of antigen-presenting cells. Again, both of them use the major histocompatibility complexes. Hopefully I've written this out enough times now where I can just start abbreviating it MHC to express some non-self antigen. It could be non-self because it is from a pathogen. It could be non-self because it is an abnormal protein that is being made by a cancer cell. It could be a non-self antigen because it is viral. It could be non-self antigen because it is something foreign from another person's body that has been put inside of yours. For whatever reason, it is expressing that non-self antigen. And I said there are two main types of antigen presenting cells. And if you also remember, I said earlier that there are two main classes of major histocompatibility complexes. And so that's not surprisingly because both types of antigen presenting cells express a different type of major histocompatibility complex. All right. Let's talk about those. Talking first about the class 1 major histocompatibility complex. Now, there's a couple keys to this. Let's do this way. All right. This here is a normal healthy cell of your body. Okay? As we have talked about, job of your cell is to make proteins. Those protein pieces, right, the antigens, are picked up by class 1 major histocompatibility complexes and expressed on the surface of the cell. All right. This is kind of what we've already kind of talked about. This cell inside here is going to make a bunch of major histocompatibility complexes. And these major histocompatibility complexes are going to, oh, that's horrible. Grab proteins from inside the cell. And these things get expressed on the surface of the cell. Now, if this is a normal, healthy cell, this is a normal, healthy us antigen. And should that antigen stimulate any of our T cells?
No.
No, because remember, our T cells that are floating around inside of our body are immunocompetent T cells are self-tolerant. So they shouldn't respond to this, and everything is fine and everything is dandy. However, what happens if this cell becomes infected? If this cell becomes infected, then as we know, what happens is that our cell is now hijacked. And it is making new viruses. And in making new viruses, it is making viral proteins. And as it's making viral proteins, that means that viral antigens will be grabbed by the major histocompatibility complex, class 1, and expressed on the surface. All right. So what's going to happen now is the major histocompatibility complex. Oops, hold on. Our major histocompatibility complex. Now, when it's grabbing some random protein from inside of the cell, happens to grab one of these viral antigens. And so now, when it is expressing itself on the surface, it is expressing a foreign antigen, something that isn't us. So notice now, this particular class 1 major histocompatibility complex has both a self and a non-self component. And this is what we mean by a double recognition. So that when our T cell comes in, and it has the right shape binding site to be able to bind to this class 1 major histocompatibility complex, recognize both the self, recognize the non-self, and get that double recognition. So notice what's happening here. We have our class 1 major histocompatibility complex, and it is expressing an internal protein. By internal protein, I mean a protein that was made inside of the cell. It is expressing that on its surface. And when it does that, it is going to activate our cytotoxic T cells. So we're getting this double recognition that is occurring due to an internal protein that is being expressed, right? So notice, remember, an infected cell is going to be expressing abnormal antigens, things we don't recognize because they're viral. Remember, we also said how this would work for, oops, I didn't mean to make that pink, I don't care. This would also work for cancer cells. Cancer cells make abnormal proteins that it's not going to be able to recognize. This would work for foreign cells, right? Because a foreign cell from somebody else, right? Someone else's liver comes in your body, they make different proteins than you do. And when they express those different proteins on the surface, that could stimulate an immune response. So again, since we're talking about the immune response, we primarily think of it in terms of an infected cell, but it can be other types of cells. And that's because almost all, most cells of the body express these class 1 major histocompatibility complexes. So most of the cells of the body have these class 1. So when they're infected, when they're abnormal, when they're cancerous, we can recognize them.
So this blue T cell that you just
Yep.
Is the cytotoxic T cell?
Yes. I put a big K in here because it's the killer. Cytotoxic T cells.
Okay.
And luckily, we'll have a way to identify those. We'll give them a fancy name in just a minute.
So is the selection of the protein just kind of a random process for the cell that's infected where they're throwing up either their own or a random one that's not?
So yes, so great question. So there's two aspects of this that I want to address. Remember, what's happening inside of the cell is these major histocompatibility complexes are being made, and then they're being expressed, and then they're being brought down. And a new one is made and put up, and then a new one is made, is put up and brought down. And so it is a constant process that the cell is doing. So you're right. What protein it grabs and expresses is totally random. But if it's a healthy cell, then all the protein pieces that are inside the cell are all things that are that are not going to stimulate our T cells. But if it's a cancer cell making abnormal proteins or an infected cell or some foreign cell, then yeah, occasionally the pieces it's going to grab is going to be something that isn't us and will stimulate a T cell. And notice in this case, it stimulated the T cell that had the V-shaped binding site. What determined whether or not you had a T cell with a V-shaped binding site?
Your genetics?
There you go. I'm going to keep beating this dead horse until you guys knee-jerk reaction to it. Genetics. Whether you have that V shape or not, to recognize this particular viral antigen is determined genetically. All right? If you don't have that V shape, it becomes a much harder process. We can still fight it, we can still recover it, but it's a harder process. All right.
So would that explain why some people are prone to getting cancer and some people never do?
No, this doesn't affect your body's ability to get cancer or not get cancer. What this would influence is your body's own personal defenses being able to attack cancerous cells, right? Our own body's defenses are able to attack some cancerous cells. It depends on how abnormal the proteins that are being made from it are. Cancer tends to grow very rapidly and very out of control, so it's typically not one of those things we can rely just on our own immune responses. And so that's why we do other things like, cancer and, targeted treatments and things along those lines for cancer. So it doesn't affect the likelihood of you have cancer, but it could influence how effective your own immune system could be at helping you to defend yourself against it.
Okay, so if your immune system is genetically programmed to be better at defending against cancer cells, then would that make you less prone to getting cancer? I guess is my question.
It wouldn't stop you from getting cancer. What it would do is make your body more efficient at attacking cancer if you got it. But really the problem with cancer is you got the production of abnormal proteins. And my concern with that would be that while it's producing abnormal proteins, the proteins probably aren't super dissimilar to your own proteins. And so my concern is if you had an immune system that was very good at attacking the abnormal proteins of a cancer cell, that might actually increase your likelihood of getting an autoimmune disorder, because it might make it more likely that your immune system would be more likely to react to your own proteins, your normal proteins as well.
Okay, so it's like a balance.
Yep. All right, let's go back to the pretty picture. So again, here we see that example. Notice our class 1 major histocompatibility complex. In this case, we're making viruses. And as we make viruses, a piece of that virus is attached to the class 1 major histocompatibility complex, and it is expressed on the surface. Right? Normally, it's expressing our normal pathogens, our normal antigens, but in this case, it's expressing a piece of an virus. So again, notice in this case, it is an internal antigen. It is something that was made inside of the cell. And that's the key. Class 1, and I think of it 1 as an I, because they use the Roman numerals, I for internal. It expresses an internal protein. We'll talk about what happens when the T cell binds to it in just a second, because there's more to it than just that. Okay. No, we'll talk about the activation of the T cells in a moment. Let's build to that, because there's more involved. All right. Think of it this way. If you put your key into the ignition of the car, I know back in ancient times, there were things called cars that ran on gas. And prior to that, they actually had keys, right? You didn't just have a fob that you kept in your pocket and you waved your hand and the car magically came on. There was once a time where you actually had to insert a key into the ignition of the car. And when you inserted that key into the ignition of the car, did the car instantly start up? No, that would be dangerous. And it's the same thing with our T cells. We want more than just the key in the ignition to start up that T cell. We need more than that. So we'll get to that in a minute. Let's build. All right, let's talk about our class 2 major histocompatibility complexes. These are the ones that are expressed by phagocytes and also by B cells. And they are presenting an external antigen, right? If you think about it, this makes sense with our phagocytes. Phagocytes take something from outside, gobble it up, and then express it. So notice it's not a protein they made. In this case, it is a protein they engulfed. right? Let's switch back to the board and play with this again. So again, is it worth trying to erase little bits of this? No, probably not. Let's just go ahead and erase the whole thing. So here is our phagocyte. As we know, our phagocyte comes in contact with a pathogen, gobbles that pathogen up, endocytoses it, uses the lysosome to break it up into little bits. So here's our vesicle. We break it up into a whole bunch of little bits. And here in this vesicle, because it was on the surface at the time, we have our major histocompatibility complexes, or when it binds with the lysosome, that lysosome may have the major histocompatibility complexes. And so we have this major histocompatibility complex, and this major histocompatibility complex can bind to one of these antigens from when it broke it down. And so it grabs onto one of the antigens from this broken down pathogen. Then, as we know, this comes to the surface and is released via endocytosis, where most of the material is released. But when that vesicle is binding to the plasma membrane, that expresses that major histocompatibility complex class 2. Oops, that is not what I wanted. Actually, I'm going to cheat and move it up here near the top. It's going to express that major histocompatibility complex holding that external protein. Right? So we ingest a pathogen, and then we express that external antigen on the surface. So notice this is one that it didn't make. This is one that it found. And that's really kind of the big key with these. We'll go back to this in a second. Class 2, major histocompatibility complex, express external antigens. And again, the cells that do this are phagocytes. And as I also said, our B cells, as we will see. All right. Questions on this? Yes, so when the cell phagocytoses, it then expresses. And maybe, again, my artistic skills are not the greatest. So here's the pretty picture from your textbook. Notice here we have some vesicles inside. We have the endocytosis of a pathogen. That formation of the phagolysosome, where we break down the pieces. And notice here's another vesicle with a class 2 major histocompatibility complex and a placeholder. When it binds to this vesicle, it is able to grab onto one of those antigens and express it on the surface. Remember, there is a key to this. If you think about this process, when these class 2 major histocompatibility complexes are expressing an external antigen, is this cell saying, kill me? Is this phagocyte saying, kill me? No, the cell is saying, look what I found. This is going on, this thing is floating around inside of our body, go kill it. So basically in this case, it's telling the T cell to go kill this thing I found. All right? This is something I found. It's inside of our body. Go kill it. But what about the previous one? With our class 1 major histocompatibility complex. Notice this is a cell that is making this protein and expressing it on its surface. When it's expressing it on its surface, what is this cell telling the T cells?
It's infected.
Yeah, I don't belong here. Kill me.
Kill me.
Right? So that's basically what it's saying. So the class 1 major histocompatibility complex is expressing an internal protein or antigen that it made. And basically it's saying, look, I'm making this weird proteins. Something is wrong with me. I'm infected. I'm cancerous. I don't belong here. And basically what it's saying is kill me. I'm not making the right types of proteins. You need to kill me. Class 2 is saying, don't kill me. Go kill this.
So the class 2 MHC, it takes the not digestible thing that like was left after the phagocytosis.
Yeah, remember when we use that phagolysosome and we break down the pathogen, we destroy the pathogen, we destroy its DNA, we destroy its protein capsule. And we take pieces of that protein capsule, which have its antigens on it, and we express them on the surface of our phagocytes to say, I just gobbled this thing up. Look what I found. Right? It's when that cat finds the mouse in your house, kills the mouse, and brings it to you. Look what I found. Right? You don't kill the cat when that happens. That's your cat way of telling you, yeah, I killed this, and this means that if this is running around the house, there's probably 5 more. And that's what's happening with these class 2. All right, so both express different types of antigens and both give the T cells a different type of message. Either go kill this or kill me. All right, questions on that? All right, excellent. Now that we know about our antigen presenting cells and the 2 types that are going to activate our T cells, what we need to do is talk about the activation of the T cells and how that actually occurs. All right. Again, this continues to get deeper. This is a good stopping point. So let's go ahead and take our first break here. I see that it is 1:17. So let's come back at 1:32. And at 1:32, we will pick up the lecture from here. Any questions before we take our first break? All right, see you in 15 minutes. All righty, let's go ahead and get started again. Now, it's always weird with this whiteboard. Oh, don't want to do that. All right, can you guys see all of these boxes or just at least the top of them?
Top of them.
All of it? Excellent. Okay, perfect. All right, so we know that there are 2 different types of cells that express 2 different types of major histocompatibility complexes. Excellent. Some cells express the class 1. Let's move this button. Yeah, let's make it smaller. Class 1, major histocompatibility complex. Remind me again, what type of cells express the class 1? Most cells, true. Okay, you are correct. Most cells. But for our immune response, whoops, what kind of cells are we typically thinking of? Infected, there you go. These are infected cells that express it. And again, most express them normally, but normally they don't facilitate a response. When they facilitate a response, it's an infected cell that is doing them. So let's go ahead and do infected cell for now. What type of protein is it expressing?
A foreign one?
True, a foreign one, but is it one that it made or one that it collected from somewhere?
It made it.
Right, an internal antigen. Excellent. Over here, we have our class 2 major histocompatibility complex. What type of cells typically express these? There you go, our phagocytes. And excellent, you guys are anticipating my questions. I like that. Phagocytes. Phagocytes, and it expresses external antigens. Perfect. Excellent. So here we have our two classes, and let's go ahead and give them their foreign antigen, and we'll just make them squares because they're easier to draw. All right. Now what we need to do is we need to talk about our T cells. T cell activation requires a couple of things. All right. The first of this is that double recognition we talked about. That double recognition is that ability to recognize both the self and non-self antigens. All right. That is the first thing that our T cell that T cells require for activation. But there is a second thing that is needed as well. And to understand that, we need to understand another component of our T cells. We need to talk about the T cells themselves. I've been drawing them as these little circles. So let's go ahead and draw them as a little circle again. Here is our T cell. But this T cell has its own proteins on the surface. It has its own antigens, things that are called CD markers. And there are over 70 different types of CD markers that can be on the surface of a T cell. Now, do all T cells have all 70 plus of these on them? No, of course not. But there are some that all of them need. So for instance, all viable T cells need what is known as a CD3 marker. So this CD3 marker is a little antigen on its surface. We'll call this the CD3. And it turns out if a T cell doesn't have this CD3 on its surface, it's not able to be activated. It's not able to divide. So that's something that is pretty darn important. So pretty much any viable T cell must have the CD3 marker. But others have different ones. So different cells have different ones. And there are two really important ones. CD markers. Those two really important CD markers are called CD4 and CD8. Whoops, that's not an 8, that's an 8. So when we draw, and I'll draw two more T cells here, one over here and one over here. Let's go ahead and bring this down, that'll be fine. As we said, the first thing that our T cell needs to be able to do is it needs to be able, let's do this in green to emphasize the difference. Oops, no, wrong. Double recognition. So it has to have a receptor on it that allows it to bind to the major histocompatibility complex and bind to the correct shape of the antigen. But it also has these CD markers, and there are two different CD markers. The first possible CD marker is CD8. So we'll draw CD8 just kind of as a little linked line over here. This is our CD8 marker. And because you can either have CD8 or CD4, you can't have both. That's actually how we identify the individual T cells. Oops, hold on. I wanted that there. And then this, we can make big. So this particular cell we call a CD8 T cell. And this CD8 marker allows this T cell to bind to the class 1 major histocompatibility complex. I need this. I need that. Okay. Or you can have the CD4 CD marker. And just to make it different, we'll draw it as a squiggly line. This is our CD4 marker. And it allows the T cell to bind to the class 2 major histocompatibility complex. And guess what we call this type of T cell?
CD4 T cell.
There you go, CD4 T cell.
You typed CB4, CB, not D.
Oh, you were right. You were correct. Sorry. CD4. And just so that this isn't confusing, we'll do that. And then I can grab that and put that there. There, that better? Nope, no. Jiminy Christmas. There we go, good enough. All right. So we have that double recognition, that ability to recognize both the self and the non-self, but each different type of T cell is going to bind to a different class major histocompatibility complex. CD8 only binds to class 1 major histocompatibility complex. Ugh. And CD4 only binds to class 2 major histocompatibility complex. And the way I remember it is, if you think about it, 8 times 1 equals 8. 4 times 2 equals 8. So CD4 binds to class 2. CD8 binds to class 1.
Last thing we need to add to this is what each one of these particular cells is going to become. As we mentioned, the CD8 T cells primarily become the cytotoxic T cells, and our CD4 primarily become our helper T cells. All right. Questions on that? Said, this doesn't make sense. It's just about to get worse. So make sure you understand this. But remember, I said there are two things that need to happen to activate a T cell. And so the second thing that needs to happen, what color haven't I used yet? Please orange. The second thing that needs to happen is what we call a co-stimulation. I made the joke a little bit earlier about the key in the ignition. When you put a key in the ignition of the car, does the car instantly start?
No.
No. What do you need to do to start the car? Turn the key. Not only do you have to put the key in, but at the same time that the key is in, you have to turn the key, right? If I just sit here in my house and I turn my key all day long, my car doesn't start. If I put my key in the ignition and I leave it there all night, my car isn't going to start. It's only when the two things happen together, the key is in the ignition and I turn the key, only when those two things happen together does the car start. And that's the exact same thing that happens here with the T cell. Yes, it needs that double recognition. It has to bind to the major histocompatibility complex, either one or two, but at the same time, there must be a co-stimulation. This co-stimulation is a chemical signal released by the presenting cell. This co-stimulation can occur one of two ways. It can either be a protein expressed on the surface, or it could be a chemical released. By the antigen presenting cell. Now, I'm going to show both of these. I want to make sure to emphasize the point, right? Class 1, so here the example is, for instance, it has a protein expressed on the surface, and that is providing the co-stimulation. Or, as I mentioned, it could be a chemical that is being released. And that chemical that is being released causes the co-stimulation. Now, I'm showing both examples. By showing these both examples, am I telling you that class 1 cells always have proteins and class 2 cells always release chemical signals? No, it's one or the other. Sometimes it'll be release a chemical signal. Sometimes it will be a protein that is expressed on the surface. So those can mix and match. So here, sometimes over here, it could be a chemical signal that is being released. Sometimes over here, it could be a protein. So it's going to be one or it's going to be the other. So here, let's just go ahead and draw both. That makes it easier. When you put that key in the ignition and you turn the key, then finally the car turns on. Probably not both. I guess there's nothing saying that it can't be both, but it's probably not as common. And again, it's like we were talking about before. These are cells that are capable of destroying other cells, or T cells, or destroy other cells in our body. These are not things we want to turn on accidentally. We only want to activate them when there really is something bad and harmful in our body that we need to destroy. That's why we're really careful about which ones get matured. That's why we're really careful about turning these things on, so that it doesn't happen by mistake. Now, again, if you don't like my pretty pictures, let's take a look. Well, we'll talk about why CD4 is so important for HIV a little bit in this class. We have a group who is presenting HIV, so I don't want to steal too much of their thunder. But yes, we will talk about that. All right, let's go ahead and go back to the pretty pictures. So remember, we don't want to have that inappropriate activation, so we're going to require a co-stimulation as well. Right? This is separate from the major histocompatibility, and it can either be a plasma protein or some type of chemical signal. All right? So as we mentioned, our T cells have even more special plasma membranes on their proteins on their plasma membrane. The CD markers, over 70 of those. CD3 is what's necessary to activate the cells, but some will have CD4, some will have CD8. No new information here. This is everything we just talked about before, but this is what I wanted to get to. Here is the pretty picture. So notice, here is our class 1 major histocompatibility complex. And remind me again, because again, I want to beat this dead horse to make sure everybody understands it. What cell is the antigen presenting cell in this case? This would be an infected cell. Right, that it's an infected cell expressing that major histocompatibility complex, class 1. And what type of antigen is it expressing? An internal antigen. Excellent. It is presenting an internal antigen. So there's the internal antigen on that class 1 major histocompatibility complex. Here is our T cell. And this is a CD8 T cell because it has the CD8 protein that allows it to bind to this class 1. So that is step 1 in this process. We get this double recognition of both self and non-self. But at the same time, we are getting that co-stimulation. That chemical signal that is occurring at the same time that is going to activate this T cell. And most of the T cells that are activated this way, most of the CD8 T cells become cytotoxic. Of course, I keep saying that, most, almost all, but most is not all. So those others are going to become our memory cells and also our suppressor or regulatory T cells. So most become cytotoxic, but we will produce some memory cells and we will also produce some regulatory T cells. All right. Again, no new information, just the pretty picture from your textbook. Here is our CD4 T cells. Notice they have a different shape CD4 protein that allows it to bind to our class 2 major histocompatibility complex. And again, what antigen presenting cells express the class 2? Phagocytes. And what type of antigens are they expressing? External. Excellent. So phagocytes expressing external antigens. And notice our pretty picture that our artist has made for us here has forgotten the co-stimulation. Remember, we also must have co-stimulation at the same time. Both of those things must occur at the same time. That double recognition of self and non-self and the co-stimulation. And when that occurs, these CD4 T cells become helper T cells. 4 for CD4, 4 for help. These form the helper T cells and are also going to form some memory and some regulatory T cells.
So if there is no co-stimulation, can it be inappropriate activation?
It can. So what happens in that case is the T cell becomes what we call sensitized. And yes, unfortunately, what can happen is a sensitized T cell can be inappropriately activated. And that can be one of the things that can potentially cause an allergic reaction, a hypersensitivity, an autoimmune response, yes. All right, questions on that? Good news is it gets easier from here. All right, now we know how to activate our T cells. And now that we know how to activate our T cells, and again, we can talk about what happens to our T cells when they're activated. The first thing that happens when our cells are activated, and it doesn't matter whether it's a CD4 or a CD8 T cell. In both cases, we are going to get rapid cellular division, producing a massive number. Wow, that's way off. Producing a massive number of identical cells, right, that are specialized for that same pathogen, right? And that's the key. Remember, we talked about this being specific. They're all going to be specialized for the specific antigen that activated the original one. It allows us to be very strong in our response and very precise, very specific in our response. So we get the production of many, many identical daughter cells, most of which become activated. There's that pesky word, most again. Those that are not activated, what do you think happens to them? Exactly. They become the memory cells. And those memory cells don't help in this response. They don't help with this infection. Instead, they make a second defense to the same infection, to the same pathogen, a stronger response. So they don't help us on this one, but they're there to help us in the next one. So most of the T cells are going to be activated, but a few of them stay unactivated, stay inactive. And they're the memory cells that are going to help to protect us in the future. All right, questions on that? All right, there you go. Some stay inert, become memory T cells. So let's talk about the cells when they are activated. Here we see again that pretty picture of the activation. We have a T cell that has a receptor to bind to both the class 1 major histocompatibility complex and the antigen, the self and the non-self. And it's receiving the co-stimulation from the antigen presenting cell. And as we mentioned, the first thing that happens is it divides rapidly, producing a massive number of daughter cells, most of which are activated, but a few of them become memory cells. And our cytotoxic T cells, as we talked about, are the killer T cells. Their job are to kill any cell that is expressing that antigen, including the one that activated it, right? Remember, when a cell is expressing that internal antigen, it's saying, I'm making this, there's something wrong with me, kill me. So these cytotoxic T cells are going to find that cell and any other cell that is expressing this antigen and kill it. Now, remember, we said natural killer cells were modified lymphocytes. So would it surprise anybody if our cytotoxic T cells use the exact same types of methods to kill a cell? It wouldn't surprise me, right? These are the only T cells that can directly attack other cells. Only those displaying the class 1 major histocompatibility complex and that specific antigen. So again, it can be parasites, infected cells, cancer cells, transfused or transplanted cells, whatever it is. We've said that a couple times at this point. And like I said, it's going to use a similar method to what we saw in the natural killer cells. It can get the cell to lyse by releasing a special protein that is going to poke a hole in the plasma membrane, what we call perforin. Also, that perforin is going to allow other proteins that are released, like a lymphotoxin, that is going to destroy the cell's DNA and induce that programmed cell death. And what was the fancy word we used for that programmed cell death? Apoptosis. Excellent. So it is going to trigger that apoptosis. And then it's going to move along to the next cell and do the exact same thing. Right? So it can use lymphotoxin to destroy the DNA, and it can induce that cell death by releasing special enzymes called granzymes, which trigger that apoptosis. And your book's got a pretty picture that shows this. Oh, I should say that too. Remember, we also talked about how our cells, our infected cells, will release those interferons. And those interferons, remember, protect surrounding healthy cells and also stimulate phagocytes to come to the area, macrophages to come to the area to destroy the cell as well. So it can also stimulate interferon release. Again, I like the pretty picture from your textbook, so here we see. Notice our killer T cell comes and binds to it, releases those vesicles, pokes the hole in it, releases those granzymes, those lymphotoxins, destroying this target cell, and then it can move on to the next. All right, questions on that? All right, things get a little bit more interesting with our CD4 cells. What do CD4 cells become again?
Helper.
Helper T cells, exactly. These are our helper T cells. Again, here we see the activation, and once again, notice they peskily have forgotten our co-stimulation on this picture, but we know it's there. And just like with the cytotoxic T cells, when activated with that double recognition and the co-stimulation, the first thing that happens is cellular division, producing massive numbers of cells, a few of which stay inactive, become memory cells, but the rest become active helper T cells. And helper T cells, as the name indicates, their job is to enhance and coordinate the entire immune response. And really, you could say the entire body's defenses. Now, if it's going to control all of these things, how's it going to do it? It's got a big megaphone and it yells supportive things. That was sarcasm. How does it communicate? There you go. Chemical signals. Absolutely. These cells produce and release a massive amount of chemical signals. And as you guys remember, as we talked about, these are a major class of hormones or hormone-like proteins. Or hormone-like proteins called cytokines. I know when you get to micro, you guys will learn about all the different types of cytokines and all the fun things that go along with that. I'm not going to worry about distinguishing and making you know all of the different types. I don't have a good place to put this. So let's just do this. Oops. Coordinates the immune response by releasing, again, a big, huge class of hormones and hormone-like signals called cytokines. When helper T cells release these cytokines, they do all sorts of things. In fact, one of the very first things they do is they stimulate the production of more helper T cells. So one of the first things they do is make more helper T cells. The first thing they stimulate is they stimulate themselves. We call those autokines because they affect themselves in that fashion. They stimulate cytotoxic T cells, maturation. So not only does it cause more to be made, but they also mature faster to stimulate a stronger response. They also play a very important role in enhancing our non-specific defenses. They can attract phagocytes to the area. They can attract natural killer cells to the area. They can enhance inflammation. They're the ones that release the pyrogens, right, that cause fever. And if that wasn't enough, they also are responsible for the activation of most, so let's say it this way, required to activate most B cells. There are some B cells that are self-activating, but most B cells actually need the helper T cells to be activated. After all, if you remember, I mentioned how it is the T cells, the cell-mediated immunity that is activated first before the humoral. And that's because humoral, which requires the B cells, most B cells require the activation from helper T cells. They can't activate on their own. This is a lot and super important. We got a pretty picture that does a nice job of showing this. Again, notice here we have our helper T cell, double recognition, co-stimulation activated. As a result of that, it stimulates itself, it stimulates the cytotoxic T cells, it stimulates the B cells, and like we said, it enhances the non-specific defenses, making these helper T cells vitally, vitally important. Which is why it's a really good thing that nothing ever hurts our helper T cells, right? Because if something hurt our helper T cells, we might be in a lot of trouble. But nothing affects our helper T cells. Anybody got a problem with that statement?
Probably an autoimmune disorder.
Oh, maybe. Although one group in particular should definitely be aware of something. A little concerned. Is there something that affects our helper T cells? Anyone? There you go. I would prefer those question marks not be there, but yes, you guys have hit it on the head. HIV is a disorder that specifically targets and destroys helper T cells. Someone earlier mentioned why with HIV people monitor the T cell numbers, because that's what it's attacking. It's attacking your T cells. And if it's destroying your T cells, what happens to your ability to build up an immune defense? Yeah, it goes to pot, right? And again, like I said, I don't want to steal too much thunder from the group that is presenting HIV, but most people who have HIV don't die from HIV. What they die from is what is called an opportunistic disorder. Something like the common cold, which normally you have no problem fighting off. If the HIV virus has destroyed your helper T cells, you're not able to get and stimulate and enhance your immune response, and something that normally a healthy person could fight off can easily kill you. All right. I would have, until about 6 months ago, argued that HIV was probably the most dangerous virus that had ever been discovered in the history of man, right? Now it's got a new contender. We'll see what happens with the good old beer virus. But for now, again, we have an understanding of how this works. All right. Questions on that? All right, here is a pretty picture that shows this process. Again, the CD8s can become cytotoxic memory or suppressor or regulatory. The CD4s become helper memory and also suppressor. I know they don't have this. It's been interesting. In the time that I have been teaching anatomy and physiology, which granted has been 150 years, but in the time that I have been studying this, our understanding of these suppressor or regulatory T cells has changed dramatically. At first, it was believed that just the CD4s formed the regulatory cells. Then it was believed that just the CD8s turned the regulatory cells. And now it is understood that they actually both do. Both CD4 and CD8 can form suppressor or regulatory T cells. But that's kind of the issue. These things are very, very hard to study. It's hard enough to study the immune response and see and understand how it works. Just look at how much we're struggling with that right now in trying to fight the coronavirus. And that's something very simple and straightforward. How the regulatory T cells work, the interactions, the chemical signals is even harder to understand, but it's definitely something that is important. The regulatory or suppressor T cells are the ones whose job is to shut down the immune response after it is no longer needed, right? You rally the troops, you build up this big, huge, massive army to be able to fight against that, whatever that harmful thing is. But as soon as you've destroyed that harmful thing, do you want all these cells riled up and ready for a fight roaming around inside your body with nothing to do? No, they might get bored and they might start attacking things, right? In fact, there are some, I believe that the research is showing less and less that this is not likely. But at first, when they were looking at the tremendous increase in allergies that was occurring in people, there was a theory that what it was, especially because it's so disproportionately increasing in first world versus second or third world countries, is that the problem were things were too clean, right? Because we kept the environment so sterile, all of our kids' immune systems got bored and so just started attacking the body because it didn't have other things to attack. Right now, again, it doesn't appear that that is holding as much water as we thought. It may play some role in it, but there were some who thought that that was the primary thing. But that's the problem. We don't want these cells just roaming around causing trouble. So we need a way to turn them off. And that is our regulatory T cells. Like I said, it is very poorly understood. We know, like helper T cells, and this is why they originally thought it was just the CD4s, they primarily turn off the other T cells by releasing inhibitory cytokines that slow the production, slow the division, and actually can cause the death of these immune cells. Now, it's hard to study, but it is incredibly valuable. And there's a tremendous amount of money that is going into the research of this. Because if you think about it, right, this could be very, very important for people who are trying to get transplants, right? We talked about blood is pretty easy. You can get blood from a person because there's only 2 antigens that cause a severe reaction. With a liver, it's closer to 30. So it's a lot harder to get I wouldn't say it's disproven, but I would say that it has been shown that there are other factors involved besides that. So I would not say it's been disproven. It still may be a factor, but I wouldn't say that it's the primary cause. But a liver can have like 30 different antigens that can cause a severe reaction. And so it's very challenging to get a heart or to get a liver or get a lung. And especially since those organs are so valuable and so viable, you can't wait for years waiting to get a new heart necessarily. So if we were able to selectively suppress the cells associated with a new liver or a new heart, right, then think of the impact that would have for transplants, right? You could pretty much put anybody's heart into anybody else's body as long as it fit, and it would be fine, right? The problem now, even with, if you can get that liver that is pretty close, when you give that person that new liver or that new lung or the new heart, that person is on immune suppressors for the rest of their life. Because basically we need to suppress their immune response so that their body doesn't attack that. And the problem is, if you're suppressing their immune response, yeah, their body doesn't reject the heart, but it also makes it a lot harder for them to fight the common cold. Right? So if we could selectively shut off certain parts, that would help with transplants, that would help with autoimmune disorders. Again, not to steal somebody else's thunder, but type 1 diabetes is because the immune cells attack and destroy the cells of the pancreas that produce insulin. If we could go in and turn off those specific T cells that are destroying the cells of the pancreas, you could essentially cure autoimmune disorders, or at least stop them from having long-term impacts. So it is a very challenging area, but there's a tremendous amount of effort and research and time and money going into our understanding of this because of the implications in the long term. All right, questions on that? All right, so that is our cell-mediated immunity and everything that goes along with that. What we need to talk about next is our humoral immunity. This is a good natural stopping point. It's a little bit early, but this is a good natural stopping point. I think we are still doing good on time. Let me take a quick peek at the rest of the lecture, see what we have to cover. Where are we? We are here. Oh, yeah, we can do that. Okay, excellent. All right, let's go ahead and take a full 15-minute break, come back at 2:30, and at 2:30 we will pick up from there. All right? Any questions? All right, let me bring the screen back up there. All right, I'll see you guys in 15 minutes. All righty. Let's finish up. We have talked about cellular immunity. It is time to move on to our humoral immunity. And again, as we mentioned, our humoral immunity is the one that uses the B cells. Now, remember, humoral immunity we also have talked about as being antibody-mediated immunity. So when we talk about antibodies, we've been drawing them since the first section of the class as these great Y-shaped molecules that have some kind of fancy shaped binding site, like a V or something along those lines at the end of them. And we've been doing that for simplicity purposes, but if you look closely at what a real antibody protein chain looks like, you can see that it isn't that dissimilar from what we've been doing up to this point of time. It is comprised of 4 different strands of amino acids. And there are 2 light strands that are the smaller strands that we see here and here. And 2 longer strands, and for simplicity, I will just draw over them. Oops, hold on. Draw over them this way is like that as well. So there are 4 strands of amino acids that come together, 2 heavy chains and 2 light chains that form that Y-shaped structure. However, there are also 2 distinct regions to the antibody as well. Basically, everything here that is emphasized as being lighter, and let's go ahead and do this like this. So basically, this part back here is the constant region. The key to this is that every single antibody that your body makes has this precise shape to it. Remember, we've talked about this being a flag, the end of the Y being a flag that gets waved around. Something easy for a phagocyte to be able to grab and to hold on to. And the reason it's able to do that is because every single antibody in your body all have this exact same structure to this constant region. So when we've been drawing our Ys to our antibodies have always looked the same. What hasn't looked the same is the binding site. That binding site, and we'll do the binding site in blue. There we go. Is this portion up here, or this portion up here. And this binding site is the part that either has the V shape or the circle shape or the triangle shape or the square shape or whatever shape it is. And what determines what variability you have to your binding sites of your antibodies?
Genetics.
Say again?
Genetics.
Genetics. Yeah, genetics. Your genes, absolutely. Your genes are what determine what the shape of those binding sites are going to be. Now, one other point to emphasize is notice every time we've drawn these Y shapes, we've always put the same binding sites on both sides, and that is the case. You're not going to get a triangle on one side and a circle on the other. So every antibody subunit has two binding sites that are going to be identical to each other. All right? So there you go. And again, always determined by your genetics. Now, that is the general composition of an antibody, or again, an immunoglobulin. Those two terms can be used interchangeably. But there are 5 major classes of immunoglobulins that have different and specific functions. So let's identify those as well. The first, it really is just the heavy chain because it has more amino acids in the sequence. So yeah, if you go back and look at it, there are more amino acids in the heavy chain. So they could have just as easily been long chain and short chain, but they used heavy and light. So I don't know why. Mostly because anatomists hate you would be my guess. Because long and short make more sense, but they went heavy and light. But yeah, that's the only reason, because the number of amino acids. All right. The first major class of immunoglobulins are what are known as IgMs. Ig, of course, standing for immunoglobulin, and M is the class. These are the ones that are most rapid to respond to initial injury. These are also the very first ones we came in contact with when we were talking about blood typing. When we were talking about blood typing, remember when we added antibodies to our unknown blood, we looked to see if it would attach to those blood cells and clump them together, a process we called agglutination. So those antibodies that are actually used for that agglutination for that test are IgMs. These are also the ones that fix our complement protein in the classic binding process, as opposed to the alternative. However, we have also talked about immunoglobulins that are able to be released from the body in our secretions. And in the case of breast milk, be passed on to our infants. Right? Those are our IgA immunoglobulins. These are not only expressed in breast milk, but they're also expressed into most of the mucous membranes of the body. Their job is to help to keep pathogens out of those cavities. So they're found in most mucus secretions, so the nasal cavity, the oral cavity, urethral, vaginal, anus. These are openings to the outside world with glandular secretions, and of course breast milk as well. So these are the ones that are passed on from mom in that naturally that passive, naturally acquired immunity. We haven't talked about the activation of B cells yet, but to activate the B cells do require a special class of immunoglobulins. These class of immunoglobulins are IgDs, and these are actually going to be expressed on the plasma membrane of our B cells. So these are expressed on the plasma membrane of the B cells, and it is this expression of these on the plasma membrane of the B cells that actually help them to be activated. So we'll talk about that activation process in just a moment, but it's these IgDs that help us to do that. The most common immunoglobulin is IgG, making up about 80% of the antibodies we find in our body. And so obviously these are the ones that are primarily involved in our immune response. And both the primary immune response, the first time we are exposed to something, and also the secondary immune response, when our memory cells help to protect us from something that we have already been exposed to. These immunoglobulin Gs are also the class that are capable of passing the placental barrier. So we talked about how, for instance, with an Rh negative mom having an Rh positive baby, there's the risk that mom's antibodies can actually cross the placenta and get into the baby and damage the baby's blood. Right? We talked about the need to take RhoGAM, for instance, to be able to suppress that. That is because it is these immunoglobulin Gs that are able to cross that placental barrier. And last, but definitely not least, is our immunoglobulin Es, IgEs. These are the ones that play an important role in mediating inflammation. These are the ones that actually their job is to bind to our mast cells and bind to our basophils. And when it binds to those basophils and mast cells, and then that antigen is present, that causes the release of histamine. So it causes the release of histamine. This is very important to stimulate that inflammatory response when we need that inflammatory response. But that also means that these IgEs are the ones that are responsible for inappropriate histamine released from that hypersensitivity. And these are the ones that can lead to that anaphylactic shock as a result of that inappropriate release. So hypersensitivities and allergies and those extreme reactions we can get from those are also caused by our immunoglobulin Es. All right, so there you go. Those are the 5 of them. If you have trouble remembering them, just remember good old Madge, right? You met her at the local diner. She gives you chicken fried steak on Sundays, right? And those are your M's, A's, D's, G's, and E's. All right, now, these are the different classes. And remember, for each one of these, those Y-shaped antibody subunits are the same. However, there is 2 big differences in these immunoglobulin classes. When we look at them, it is their arrangement. Most of them, the G's, the E's, and the D's, are a single subunit. A single Y-shaped structure. And remind me again, how many binding sites does that Y-shaped structure have?
2.
2 total. Excellent. Absolutely. However, our immunoglobulin A's are actually a dimer, where we actually have 2 subunits bound together. Remember, the immunoglobulin A's are the ones that are released into the secretions of the body to capture and immobilize pathogens before they get a chance to enter into our body. And how many binding sites does this unit have?
4.
4, absolutely, it has 4, allowing it to be able to grab things, clump things together into a bigger mass to stop the spread and stop those things from getting inside. But remember, the antibody that provided us with that agglutination of the blood was the IgMs. And it turns out the IgMs are actually a pentamer, where we have 5 total subunits bound together into a single functional structure. And if I have 5 subunits, how many binding sites do I have? 10, absolutely. So you can see how easy this IgM it would be for it to grab onto multiple blood cells and clump those blood cells together, forming that big, huge mass, right, that clumps the blood together. But it also does that with the pathogens as well. Grabs a bunch of pathogens, clumps them together, right, stops them from spreading, becomes a big, obvious thing that can be easily identified and gobbled up by our phagocytes and everything else that goes along with that as well. We've kind of talked about what antibodies do, but we will get to that in a little bit more detail, give the technical terms for most of the things that we've talked about as we talk about that in just a minute. All right, so those are the 5 types of antibodies that can be produced and their specialized shapes. Questions on that? All right, so let's talk about our antibody-mediated or humoral immunity. All right, again, it uses the B lymphocytes, and our B cells carry antibodies on their surface. All right, so notice here we have a B cell, and we have this B cell, and it has basically 2 things on it. It has a major histocompatibility complex on it. And what class major histocompatibility complex did we say our our B cells have? So this is a B cell. What class major histocompatibility complex did we say that it had for it?
2.
2.
There you go, excellent. So a class 2 major histocompatibility complex on its surface. And that means that it is going to express what types of proteins again?
External.
External, excellent. So when this B cell expresses it, is it telling the T cell to kill it?
No.
No, it's saying this thing is out there and we have to do something about it. Exactly, right? So it's expressing that external, expressing the external antigen on that class 2 major histocompatibility complex. And the other thing it is expressing on its surface is an antibody. What class of antibody is it going to be expressing on its surface? Well, I had been paying attention 3 minutes ago when he was going through the classes of antibodies.
Includes 3 of kind.
Which ones were expressed? There you go. Someone's got it. The IgD antibody is the one that is expressed on the surface of a B cell. All right. So our B cells basically have these 2 things on their surface, all of them. And of course, every single B cell has the class 2, has an antibody, and every single B cell is going to have a different shaped binding site on it. So this B cell has this shape, and this B cell here has circles, and the next B cell has triangles. And of course, what determines what shape binding sites are on the surface of all of your B cells?
Genetics.
Genetics, excellent. Right, there you go. So there you go, genetics. So we have all of that. So this is what is on the surface of our B cell. Now, as it turns out, activation of the B cell requires 2 things as well, but it's not quite the same process as our T cell. All right, this I think is a little easier when I have a little bit more playing room. So let's take a look at our whiteboard one more time. And draw a B cell. So here's a B cell. Let's start it over here. And then we will put its major histocompatibility complex on here. And we will put its antibody. And again, it's going to have many antibodies. It is going to have many major histocompatibility complexes. And again, the key to this is all of the antibodies that are being expressed by this particular B cell are all going to be the same shape. Okay, so there's another B cell over here. And again, it's further away. That's why I'm drawing it smaller in size. That is going to have triangles on it, and another one that's going to have circles on it. But each one has its own unique binding site. Step 1 in the activation of our B cell is where the cell becomes sensitized. The cell becomes sensitized, and it becomes sensitized by coming in contact with its specific antigen. So what happens is there is some virus floating around out here in space that just happens to have the square-shaped antigen on it, and lo and behold, the B cell binds to it. Now remember, like we talked about, this is also how our vaccines work. So technically, it can just be the antigen by itself. It doesn't have to be the actual pathogen. It can be, but it can also just be the antigen, right? It could be because you were given a virus. It could be because a phagocyte broke down one of these viruses. And remember, it releases most of its contents. So while it kept some of its antigens to express on its own surface, some of those antigens can be released into the interstitial fluid and can bind to our immunoglobulin D antibody on the surface of this B cell. When this occurs, so again, step one is that it comes in contact with the specific antigen. Then what our B cell does, it engulfs the antigen. And when it engulfs the antigen, it then can express that external antigen that it brought in on its surface with that class 2 major histocompatibility complex. So what happens is that antigen is brought inside, and once it is brought inside, it is then going to be expressed onto the surface by that class 2 major histocompatibility complex. And at this point, it is sensitized. All right. However, most B cells then stall in this sensitized state. They're not capable of activating themselves. Instead, most need the help of a helper T cell. Most require the binding of a helper T cell. So that is step 2. All right, so we need 2 things. Notice these 2 things are not happening at the same time, the same way we saw with the T cells. These things are sequential. First sensitization, then activation. But what's going to happen next is our friendly neighborhood B cell is going to come in. Oops, no, that was supposed to be blue. Our friendly neighborhood B cell is going to come in. We know B cells have that special binding site to the major histocompatibility complex 2 to that specific antigen. So again, we get our double recognition that's going on here. And of course, this has to be an activated helper T cell. And when this activated helper T cell binds to the B cell, it releases its cytokines. And those cytokines then trigger activation of the B cell. And just like the T cell, when it is activated, the cell undergoes rapid division. Rapid cellular division, let's say that. And they give a fancy name for this. They call it clonal selection. Why it's clonal selection in B cells and not T cells, I don't have a good answer for you. The exact same process. The cells are dividing, producing a massive number of identical cells to the original. All right, so we get a massive number of new cells, and we'll make our B cells black, like the first one. Pink, oops, that's not black. Now, most of these are going to be activated, but there are going to be a few that are inactive. And those few that are inactive, what do those become? Come on, we're almost done. You can't fall asleep on me yet. What do these few that are inactive become?
Memory cells.
Memory cells. You think this is bad, just wait till next week when we're talking about the endocrine system. That's going to be brutal. Memory cells, absolutely. So there are going to be a few of these that are just going to become inactive memory cells. But most of them are going to grow to become these big, huge, large, massive cells. We'll just do it with a couple of them. And these big, huge, massive cells are going to have a massive amount of rough endoplasmic reticulum in them. And what do we use our rough endoplasmic reticulum for again?
Making proteins.
Making proteins, exactly. These cells become what are known as plasma cells. And plasma cells make massive numbers of proteins. And which particular protein are they going to make? What do you think they're going to make? If only there was a protein here that could help us in our antibody-mediated immunity. Yeah, I wonder what protein is made in antibody-mediated immunity. This is on, right? These are the easy questions. What protein do you think is made?
Antigen.
How about antibodies?
Antibodies.
Yeah. Makes antibodies. And do you think it's going to make just any old random antibody?
Specific.
Yep. And in this case, the specific antibody that's going to make is the one with the square-shaped binding site, because that is what is activated. So all the antibodies that it makes are all identical to the original. Maybe it's my art. Let's look at the pretty pictures from your textbook. So here we have a B cell with its class 2 major histocompatibility complexes and its IgD antibodies. And it comes in contact with this cute little red antigen. When it comes in contact with the cute little red antigen, it binds to that cute little red antigen. It engulfs it, and it expresses it on its surface with that major histocompatibility complex, class 2. And at this point, our cell is sensitized. Sensitized, but not able to do work. There are a few B cells that are capable. You don't have to screenshot a view. I take these pictures and I post them on the website. They're under the in the modules under lecture notes. They're on there, so you don't have to worry about screenshotting it. Sensitized. There are a few B cells that are capable. Once sensitized, they can undergo division. But the vast majority require that helper T cell to be activated. So that sensitized B cell floats around, and a helper T cell finds it, binds to it. And when it binds to it, that helper T cell releases all those cytokines. And those cytokines cause the B cell to undergo rapid clonal division, or rapid clonal selection, which again is a very fancy way of saying that the cell divides rapidly, producing a ton of new cells, identical daughter cells, a few of which stay inactive and become our memory cells, but most become active, grow to be these massive cells with a massive amount of rough endoplasmic reticulum, where they can produce a massive amount of antibodies. And I do mean a massive amount. A single plasma cell can produce as many as 2,000 antibodies per second, which is even better than those 8-year-old kids that Nike has making shoes. All right, that is super, super fast production. Now, do you think that kind of production can be maintained for a long period of time? No. So these plasma cells only last about 4 or 5 days, and then they basically exhaust themselves and die. But we got plenty more coming right behind them.
You said it was called clonal what?
Selection or division.
Okay.
What we have just described here is what is known as our primary humoral response. We come in contact with an antigen, and we produce antibodies to attack that antigen.
Is there any like hint in this name, humoral?
Well, again, remember, it is releasing these antibodies into the humors. The humors are the fluids of our body. So antibodies aren't attaching to or attacking our cells. They're being released into the fluids of the body to provide defense, right? Blood, lymph, interstitial fluid, saliva,
Okay.
breast milk, mucus, all of these secretions of the body, it is being released to it. It's being released into the fluids of the body. All right. Now, again, your book's got this great picture that kind of, we've talked about all of these things for the most part, but I want to emphasize them here because I think this does a nice job of reminding us of all the things that antibodies are capable of doing. As we know, antibodies can bind to the surface of a pathogen. When it does that, it can cause the complement proteins to come, and that can put that MAC on its surface, that membrane attack complex on its surface, and cause the cell to lyse. Remember, we also talked about how the C3A, right, C3b binds to the plasma membrane, causes the MAC to form. C3A is a chemical signal that attracts phagocytes to destroy the pathogen. And as we also talked about, it can enhance inflammation. It can attract, phagocytes to the area. All of those types of things. We've mentioned, and here's our friend, IgM, with those 10 binding sites, how it can cause agglutination. And agglutination is the fancy name we have for clumping cells together. So when we clump like the red blood cells together, or if we were to clump, some kind of bacteria or something like that together, it would be called agglutination. Notice precipitation is the exact same thing. It is clumping stuff together. Both of these two processes is basically just clumping things together. Clumping things together. However, as we know, anatomists like to give specific names for specific things. So technically, if the things that they clump together are the antigens, and since antigens are mostly proteins, they are water soluble. If you're clumping together water soluble things, technically it's called precipitation. If you're clumping together cells, since cells are suspended in solution and not dissolved in the solution, you call it agglutination. But again, I don't care so much that you understand the distinction between these two terms. Wow, that's huge. Let's undo that. In both cases, we're just clumping stuff together. Obviously, if you clump stuff together, that makes it harder for those pathogens to be able to attack our body. But it also attracts phagocytes to the area as well, makes it a bigger mess, makes it easier for the phagocytes to find them. Lastly, and this is the one that we haven't talked about quite as much. We talked about it a little bit when we talked about interferons. But remember, one of the things that we have to worry about with viruses or one of the things that we can do to viruses is stop that virus from binding to the cell. If it can't bind to the cell, it can't infect that cell. So we talked about how interferons cause healthy cells to produce antiviral proteins that will stop the virus from binding to it. But the other way we can stop a virus from binding to it is by attaching a bunch of antibodies on the outer surface. Notice if I have a virus like this that I put a bunch of antibodies on the outer surface, is this virus going to be able to bind to a cell and infect a cell if it's covered with antibodies? No. And so basically this virus has been neutralized. So really on this list, neutralization is really the only concept for antibodies we haven't talked about. It is a concept we've talked about, but we didn't talk about it in terms of antibodies. But antibodies are capable of doing all of these things. So this is what our humoral immunity is about. Destroying pathogens, blocking pathogens, decreasing the spread of pathogens, making them more obvious, making them easier to digest, stop them from coming into our body by clumping them together, and stopping them from infecting the cell. These are what our antibodies are doing for us. This is how it is protecting us in the fluids of our body. All right. Questions on this? You guys have been awful quiet today. We've been going over a lot of processes, right? We went over the activation of 3 different types of cells. Do you think one of those is likely to be an essay question on the exam? Absolutely. We've talked a lot about classifications of antibodies, what antibodies do. We've covered a lot of processes today. This is a lot of material, and your test is 2 weeks? Am I seeing that right? 1, 2, yeah, 2 weeks. Awesome. Okay. Excellent. All right. We've talked about how we activate our B cells. We've talked about our antibodies and the production of them. And like I said, we have described that as our primary humoral response. Our primary humoral response is when we get exposed to a pathogen for a first time. So here we are, exposed to that pathogen for the first time. It gets gobbled up by our pathogens. Our cells, T cells have been activated, and then finally our B cells get activated. Some of them become plasma cells, and those plasma cells start producing antibodies. As we talked about, this process does consume a fair amount of energy. Obviously, healing from an infection or something along those lines does require a tremendous amount of metabolism, a tremendous amount of resources, absolutely. Notice antibody production doesn't start instantaneously. There is a bit of delay from when you're first exposed. It can take quite some time before antibody production starts getting into high gear. And notice it's right about day 10, day 12 where it reaches the peak. Now notice they say units per milliliter. They haven't really given us the value because, again, the numbers, if we're talking about if each individual plasma cell makes 2,000 antibodies per second, I mean, the numbers we're talking about there are astronomical. So we just have a concentration unit of 1, and that is what they use. However, as we also talked about, these cells aren't going to last forever. They only produce antibodies at that rate for about 4 or 5 days. So eventually, the cells start to die, the cells start to decrease, and by about week 4, your level of antibodies is essentially where it was before the exposure. So it peaks at around day 12, and it's done by week 4. And that is our primary humoral response. You get exposed to something, you have symptoms 2 or 3 days, 4 days later. 2 or 3 days after that, you slowly start feeling better. And then about a month after the initial exposure, that virus is gone and you're back to where you were before. All right, that is our primary humoral response. However, as we talked about, what gives us the power in this specific immunity are those memory cells. What memory cells allow us to do, allow us to produce and let's be specific about this. Let me move this over here and make it a little bit bigger. They allow us to produce a faster, stronger, and longer response to a pathogen, to the same pathogen. So if we are exposed to that same pathogen a second time, we're able to produce a stronger, a longer, and a faster response. All right? Notice here with the pretty picture, we are then exposed to that pathogen a second time. Notice by day 1, we have already eclipsed what the number of antibodies we made the last time. And by the time we hit day 10 or day 12, we've produced 100 times more antibodies than we did before. And are they gone by week 4? Or week 5? Or week 6? No, notice, right? 2 months later, it is still higher than it was in the original initial response. Stronger, faster, and longer. And this second response is so aggressive that typically we have little to no symptoms as a result of it. It is a super fast, super long, super strong, super aggressive response that we have little to no symptoms as a result of that second exposure. Let's beat the dead horse. Here we see the same thing. Right? First response peaks day 10 or 12, gone by about 4 weeks. But if at 4 weeks you're exposed to that again, faster, longer, stronger response. Notice it doesn't help you if you're exposed to a different pathogen. You don't have memory cells to this different pathogen, so you still have to go through that same process a second time. Or if A mutates into B, if it changes, then we no longer recognize it. That's why we have to get the flu shot every year. That's why we still get a cold every year, because they continue to mutate. But if it's the same one, something that's a more stable pathogen, right, like tetanus, like chickenpox, mumps, things along those lines, they don't mutate as often. So something like a vaccine is very, very efficient. You get it as a kid, maybe need a booster here or there as an adult, but for the most part, you're good. And again, here we see it visually. Notice we have all the different types of B cells. The one that is able to bind to the antigen is the one that undergoes the division. Most of the cells become plasma cells, producing a large amount of antibodies, providing us with the protection. But there's that memory cell. And if that memory cell comes in contact with that pathogen a second time, we get a massive response, massive number of cells, massive amount of antibodies, massive amount of aggression, where typically there are no symptoms with a second exposure.
Do we make new memory cells from the secondary response?
Yes. Yep. And the illustration shows that too.
Oh, yeah. Okay.
Yep. All right. And I love this picture from your textbook. The page number might not be right because, again, I wrote that with the previous version of the textbook. But I know your textbook still has this picture. It does a great job of putting it all together. You're exposed to a pathogen. You have your innate defenses, your surface barriers, your internal defenses to try to slow things down, and then your adaptive defenses, your B cells and your T cells that work together, right, that help your T cells stimulate the B cell activation and everything that goes along with that. And then again, some nice tables that talk about everything that goes along for that. All right. Excellent. Spectacular.
I have a question.
I'm sorry. Yes. What?
I have a question.
Yes.
Hello.
I'm here.
Okay. So if you were to get like a vaccine, like an MMR as a child,
Mhm.
and your body forms the memory cells, but aren't exposed to it again for a long time, and your titers go down, if you do get exposed as an adult, would those few that are actually left still be effective in stopping you from showing symptoms of that disease, or would you have to have the booster in order to be effective?
So you hit the key on that. That titer is kind of a look at what kind of number of memory cells you have. Memory cells are very long lasting, and there are some memory cells that are capable of lasting your entire life. However, other memory cells over time may attrit. And that's what you're looking for. You're looking for that attrition of memory cells, and that's what they're looking for with that titer. They're looking, all right, how robust is your memory? And if it is starting to decline, then it is best to get that booster because then it'll boost it back up. If there has been a decrease in the number of memory cells, my guess is that you would still get a more robust response than normal, but might not be enough to mask all the symptoms, to be aggressive enough to destroy it before you saw symptoms as a result of it. All right, it's like, for instance, why when you step on that rusty nail, right, when you go to the hospital, they will still give you, often they'll give you that tetanus booster just in case, because they want to make sure that your body is able to defend itself in case there's that, that pathogen in there.
So if you were to be exposed again when the memory cells are low,
then my guess is that you would still show you would still show some symptoms, right?
Okay, but then your body would make more of the memory cells again?
Yes. So yes, then you would produce more memory cells. So if you're exposed to it a third time, you would then be more protected from that. Yes.
Okay.
Right? If you look at it, this is one of the concerns, one of the big questions we have about the coronavirus. One of the big questions they've had about the coronavirus is there continues to be cases of individuals who are getting it a second time. Right? And so there's a lot of concern. Everybody keeps talking about the herd immunity. As long as most people are protected from it, then the population is protected from it. But the problem is if our immune cells for this particular virus are not long lasting, there's been some recent studies that show that antibody levels may only stay elevated for as little as 4 weeks. after exposure, right? But again, the number of people that have gotten to exposures so far has been low, but it's been more than 1 or 2 cases. And again, that's also part of the problem with the lack of testing. So many people who come out in the news, right? And again, it's politicians, it's famous people, it's the people who have the money to get the testing, who come out and say, I've tested positive, but I don't have any symptoms. We don't, and that, you know, for mere mortals like you and I, who knows if we have it or haven't had it, because there's not the testing going on. Without the testing and knowing who's had it or who has it or who's going to have it, or how long they've had it for, or how long they've had antibodies for it for, there's still a lot we don't know about how long lasting that defense can be. So it definitely does vary from virus to virus. Part of that is the vigorousness of your immune cells. How long will those memory cells last? The other part of it, as we talked about, is the morphology of the pathogen, right? Things like cold virus, things like flu viruses are things that mutate rapidly. And that's because they tend to be more sloppy in the way that they make their proteins. Whereas other, like measles, mumps, rubella, those viruses tend to be very, very structured in their protein synthesis. And so once you've been exposed to that, by either having the measles or by getting the virus, then you're pretty much protected for life. And there are other things that, you know, measles is one that they're showing more. You know, tetanus is another one where it varies over time. If it's been more than 10 years, they're starting to encourage people now to, again, get those boosters, because they can be long lasting, but you will decrease the number that you have. Did that answer your question, hopefully? All right, you broke up a little bit there.
So with respect to the coronavirus, what's that? Oh, sorry, I think my internet is unstable.
Okay, so did that answer your question?
Yes, but I now have another one about the coronavirus because you forgot it for a second.
Okay.
So with respect to the coronavirus, then if they do actually manage to make an effective vaccine, is the issue that there might be a need for continuous boosters if the antibodies don't last?
So partially, but again, from what I've heard, and again, this is not my area of expertise, one of the commonly held beliefs by the current scientists is that it's never going to go fully away, kind of like the flu virus never fully goes away. So as a result of that, it may become a seasonal thing where people need to get injections seasonally to protect themselves from the coronavirus when it is more likely to be spread. So that could be one possibility. But like I said, the problem is I'm not sure there's been enough testing to see what the long-term implications of both exposure to the coronavirus and also exposure to a vaccine is. The trial vaccines, and again, this is one of the things that literally in the news right now. One of the things the FDA wants to do is to follow the progress of individuals who have participated in this vaccine study for 4 months, right? Because they want to not only look for, monitor what their defense is, how well they're able to maintain the defenses over those time, as well as looking for long-term potential side effects of the vaccine. And of course, because we have an election coming, our president is dead set against that and has already said that he is going to not approve that if the FDA tries to enforce those new rules. So again, we have this issue of politics getting in the way of science. But so the short answer is we still don't know these things. We haven't been giving people vaccines long enough to know what the long-term implications. Is it going to be something that's going to need to be yearly? Is it going to be a once and you're done thing? We don't know because we still haven't had the time to do that kind of research yet.
So I'm confused. Is the issue that we're developing antibodies, but they're not lasting in our bodies, or is the issue that the virus is mutating?
Both. It could be either. Now, my understanding is that it appears the virus has at least mutated once into a more easily infectable form. So from the original virus release, I guess there were 2 strands originally. One was more aggressive, but didn't infect as easily. And then the other version wasn't as severe in its effects on the body, but was much easier to infect. And at first we were seeing more of the first type, and we were seeing more of the second type. So again, it's been less than a year that this has been studied, that we've been watching this. So I don't think we yet know how malleable this virus is in being able to change. Okay, so that's one thing we don't know. The other thing we don't know is how long the memory cells provided from this will last, right? The fact that there have been some people that have gotten a second exposure to it and gotten sick a second time is something of a concern. But you also see that with chickenpox too. There are people who get chickenpox or get the chickenpox virus vaccine and then do still get sick by chickenpox a second time. So that type of thing where for whatever reason they didn't build up as much of an immunity the first time does occur sometimes. But how long that immunity will last is something that there's no way for us to know because we haven't been studying it long enough. We've only known about this virus for less than a year, so we don't know how long the memory cells to this particular virus will last. We can compare it to things we do know, because we do know things about other coronaviruses, and those things have been promising, and that was one of the reasons why there was less concern at the beginning of this, because we understood how normal coronaviruses worked. The problem is, in many ways, this coronavirus hasn't acted like a typical coronavirus. And that's why there's been so much change in the thought and change of understanding. And that's one of the frustrating things about it. It's both one of the cool things and one of the frustrating things about it. What we're getting to experience real time here is science. You are seeing true science being done. This is what science is. You make a hypothesis, you collect the data, you interpret the data, and then you change your hypothesis based on that. And that is how science works. You learn and you modify based on that, right? And we're getting to see that, but the challenge with that is people who don't necessarily understand science, right? Like our president, say that, well, Billy said X, Dr. Fauci said X, 6 months ago, and now he's saying Y. So the fact that he said X before and Y now clearly shows that he doesn't know what the F he's talking about. But that's exactly what science is. Science is about learning and interpreting and changing. And so it's really cool that we're getting to see this real time because it is a fascinating process. The problem is all of the politics that falls on top of it and the lack of understanding that falls on top of it. And so I think that's where a lot of people's frustrations come from. Normally in these processes, all the stuff that's going on now goes on behind the scenes because nobody cares about these things. Some problem pops up, science fixes it, and nobody gives it a second thought. Now we're seeing science firsthand, real life, live and in person. And I understand why people get frustrated for this, but I think that's because people don't fully understand what science is. And that's what this is. We are seeing science in action. And the problem is there's a lot of these questions we don't know the answers to yet. And that's the problem because we haven't been able to study it long enough. We can guess, we can interpret, right? They interpret the data, we can make hypotheses because that's what you do in science. But as we've seen, sometimes those hypotheses are wrong. And that's what science is. You make a hypothesis, you test it. If it tests wrong, you change your hypothesis and try something else. If you don't know what something is, that's how you have to figure it out. And that's what we're seeing. And so I don't know the answers. And nobody knows those answers yet because we're still trying to learn.
Okay, so why was it that it's coronavirus and specifically that became front and center? Like you said, if it's typically, for any other infectious disease, the science happens behind the scenes and it's not, something that we're aware of all the time, constantly. What did the coronavirus, specifically this one, became?
I think there were two things that caused this to be as different. I think we haven't had a global pandemic like this in years. I mean, the last time was the H1N1, right? The flu, the swine virus or whatever it was, which was.
I thought it was MERS, the other corona.
Oh yeah, SARS. There you go. So SARS as well. So those were the two most recent ones. But even those were what, 15 years ago, 10, 15 years ago, something like that. I think
MERS was 2012, 8 years ago.
Okay, so yeah, about 10 years ago. I think the biggest difference is I think it's two things. I think the first part of it is that I think that we have become a much more interactive society with all the social media, with all of the instant gratification, with all the TikToks, with all of it. Everything is immediate, and everything has kind of become like that, right? TV shows are now dumped with a whole season on TV because people can't wait a whole week to see the next episode of them, right? TikTok is basically Vine, where you get 10-second videos, because people don't have the attention span to watch regular TV anymore. All our news, everything, our entertainment, all of these things is instantaneous. And so we have access to instantaneous information. And so I think that is it. I think there's awareness of these things that we didn't have before. And I think that does complicate things. And quite frankly, the other side of it is that we have a party in power who strongly is against science. I think that we have our president is very anti-science, right? I think that he is created an environment and a culture where science is questioned. And I think that lack of general trust in science is something that has hurt this process tremendously. And stunned silence. I love it. Excellent. I try not to get political, but again, it's hard, especially when we're talking about these things, to see what is going on and to, again, it's not my area of expertise. When I say that I understand what's going on, I don't understand what's going on. I don't know anything about coronaviruses. I don't know anything about coronaviruses. Anything smaller than a cell, I don't believe in anyway. But I at least understand science and how it works. And so it's frustrating to see the attitude, the change in attitude, the anti-education, the anti-science bent that the world has taken in the past 10 years. And I think our president is a big part of that. All right. Get out and vote. All right. Off my soapbox. Again, I think it's impossible to talk about these things without talking about coronavirus. So I apologize if that got political. But again, I think I'm trying to talk about science and what's going on here. So at least I think it's relevant to what we're talking about. We're not like, we're not talking bones and I'm ranting about the government. All right, let's get back to the important things. This is everything you need to know for your immune response. Again, we could spend the whole semester talking about these things, and you're going to do a lot of that in micro. So a lot of this you'll get in micro and more. So this is enough for what we need for this section of this class, and I think that is fine for that. However, this is only half of what we're doing on this test. The other half of this test is going to be the endocrine system. And the endocrine system is just as hard, if not harder, than what we're looking at here. Because at least here, we're talking about cellular processes that are pretty straightforward. The endocrine system isn't necessarily as straightforward that way. There's a lot of interactions. There's a lot of interactions between these things, and that can be challenging. Now, one of the things I've done to hopefully help you to be more, I'm not sure what that's about. Oh, okay. Hold on. Ah, interesting. All right, hold on. Cancel that. Cancel this. Get rid of that. Get rid of that. Where is it? That's what I want. One of the things that I have provided you with to help you to be successful on this exam is the study guide. And this here is our study guide for the endocrine system. This is going to help you both on the lab exam, on the lecture exam, but also definitely on the lab exam. What you have here is a handout. For these handouts, you have all of the glands that you are going to be responsible for in the endocrine system. And you have all of the hormones that you are going to be responsible for. Now, you will notice in looking at these, we have things like we've already talked about before. Our kidney produces erythropoietin. Hopefully you guys remember what calcitriol is, right? Or atrial natriuretic peptide. That's a good one, right? A fair number of you on the exam had the question about how different things can modify blood pressure. And I do not think a single person got the stretch of the atria correct. If the atria stretch, how does that affect your blood pressure? Someone give me the right answer now that you had some time to look it up and think about it. Or if you didn't have that question and knew the answer, tell me now. Your atria stretch. How does your body respond to that, and how does that affect the blood pressure?
It would increase it, right?
Nope.
Decrease.
So if it doesn't increase it, what does it do?
Decrease.
Decreases. How? What does your atria do to decrease the blood pressure?
Expands.
Nope.
Isn't it about blood volume?
Oh, sorry, the atria that you said aorta.
This is the atria. You're right. Your aorta would expand. You are correct. Blood vessels would expand.
Yes.
Yeah.
Uh-huh.
But the heart, the atria of the heart, you were right. Stretching of the atria causes the blood pressure to be lowered in response, because what should the blood pressure be when it comes back to the atria?
0.
0 millimeters of mercury. So if it is above 0 millimeters of mercury and it stretches the atria, the heart has to do something to lower the blood pressure. What was the thing it did to lower the blood pressure? If only I was staring at something right in the middle of the field of view here.
NAP hormone.
Atrial natriuretic peptide.
Yeah, it released a hormone. Remember, the atria released a hormone, atrial natriuretic peptide. And now the fun begins. Notice this table has 3 columns: a target, regulated by, and a function. Let's start easy. What do you think the target of the atrial natriuretic peptide is?
Kidney.
Excellent. It targets the kidney. All right. What does it tell the kidney to do?
To release fluid of the body, like to make more urine.
Excellent. I like that. Release water, right? So that does 2 things. We release water. We have more dilute urine. But more importantly, it lowers the blood pressure. There you go. Excellent. All right. So far, so good. Those 2 things make sense?
What about the middle section?
Yeah, I'm getting to that in a minute. I just want to make sure these 2, the easy ones, make sense first. Do these 2 make sense?
Yes.
Excellent. So for each hormone that we're going to have here on the list, you need to know its target, what that hormone targets, not where it's produced. Where it's produced is over here, right? This is where it's produced. This is where the hormone goes. Remember, most of these hormones get into the blood, get into the circulation. And once a hormone gets into the blood, where can it go?
All over the body.
Everywhere in the body. Does that mean it affects every single cell in the body?
No.
No. So that's why we need to know what the specific target is. While atrial natriuretic peptide can get into the blood and go anywhere, it specifically targets the kidney and tells the kidney to release more water, making dilute urine, lowering our blood pressure. So what we need here, this is how it's regulated. What tells the heart to produce atrial natriuretic peptide?
High blood pressure.
True, it is the high blood pressure, but how does the heart know that there's high blood pressure?
Baroreceptors.
What kind of receptors?
Baroreceptors.
Baroreceptors, right? It has baroreceptors. Those baroreceptors are sensory receptors, and remember, they're basically stretch receptors, right? They stretch in response. So what that means is this is a neural regulation. It is regulated by the nervous system. It is a neural regulation, specifically stretch receptors.
Medulla oblongata.
I'm sorry?
Medulla oblongata.
Sure. Yes, yes, it is. All right. That makes sense. All right. Let's think about and look at one other example, an easy one. Hold on. Ah, perfect. Insulin. What is the target of insulin?
Body cells?
Liver.
Yeah, I would say most cells of the body. Any cells in particular?
Liver cells.
Liver and
Pancreas.
no, not pancreas. Pancreas is where it's produced. Bone marrow is not a bad guess, but no, not the bone marrow. Where else might we want to? Well, okay, let's go back. Muscles, there we go. Primarily muscles and liver, but most of the cells of the body. And what does insulin tell these cells to do? Excellent. Store glucose. It tells them to store glucose, uptake glucose. And of course, if they're storing glucose, where are they getting that glucose from?
Liver.
Out of the blood.
Blood, yes.
Right? Take glucose out of the blood into the cell. So if you think about it, another effect of insulin is to lower blood glucose levels. Right? I don't think that's too surprising information. I think most of us knew that, right? So then here comes a question here. How is it regulated? What tells the pancreas to release insulin?
Blood sugar level?
Bingo. Blood sugar levels. Blood sugar levels. Some substance in the blood. How much sugar there is in the blood is what tells it. That type of regulation we call, oh, wasn't there a name for the fluids in the body?
Humoral.
Humorals.
Humorals.
Humoral regulation. So notice humoral regulation, blood sugar levels. Questions on that one?
So it's not chemical signals. It just they already know.
It is humoral regulation. You've got the right idea. Humoral regulation is the level of some chemical in the fluids of the body. Of course, mostly blood, right? So yes, humoral regulation means that it is the level of some chemical in the fluids of the blood, primarily the fluids of the body, primarily the blood. And in this particular case, that chemical in the body happens to be sugar.
Okay.
All right, let's talk about what about calcitonin? That also has humoral regulation, but what does it help us to regulate? Anybody remember from 430?
Calcium in blood.
Calcium levels. So it would be humoral regulation by calcium levels in the blood.
The target also kidney, right?
That's one of the things that it targets. Yep. That is one of its targets. So notice we've had neural regulation. We've had humoral regulation. Let's look at one more. I'm going to cheat by first coming up here to the pituitary gland. Notice this hormone right here. What do you think thyroid stimulating hormone does?
Stimulates the thyroid.
Stimulates the thyroid. So if I were to come down here to the thyroid and look at thyroid hormones, guess how thyroid hormone production is regulated? Guess how we tell the thyroid to make thyroid hormones T3 and T4?
Hormone. is released from the anterior pituitary in response to levels.
Yeah, exactly. So notice thyroid stimulating hormone tells the thyroid what to do. This we call hormonal regulation. This is where a hormone in the fluids of the body, and of course by that we primarily mean blood, right, tells the gland what to do. And so this would be hormonal regulation. And not only would we need to say hormonal regulation, but we would need to say which specific hormone. And which specific hormone would that be?
Thyroid stimulation hormone.
Thyroid stimulating hormone. There you go. Notice, and let's go all the way up to the top. The good news is for regulated by, if you think about this, and I'll write this over here, for regulated by, so let's put a big, nice star right here. For regulated by, there's only 3 right answers. All right. It is either going to be hormonally regulated, humorally regulated, or neurally regulated. But for each, you must be specific. Don't just say that the thyroid gland releases triiodothyronine and thyroxine hormonally. You have to say hormonally via thyroid stimulating hormone. Your adrenal gland releases adrenaline. How is your adrenal gland regulated?
Neurally.
Neurally via what branch of the nervous system tells the adrenal gland what to do? Sympathetic nervous system, exactly. We'll go through this list together, but I want you thinking about this weekend. So as you look at these hormones, think of what their targets would be, think of what the functions are, think of how they're regulated. Like I said, it's okay if you don't have the answers to all of them. We're going to go through it together. But I want you to start looking at this, because as bad as today was, when we start working on this list on Tuesday, it's going to be even worse. A lot of processes, a lot of complicated physiological concepts. Now, I want you to notice one, so questions about that, because there's one other thing on this list that I want you to know. Any questions on this? All right, actually, I lied, 2 things. Notice for our anterior pituitary gland, 3, 4, 5, 6, 7 hormones. Does that mean the anterior pituitary gland only produces 7 hormones?
No.
No, there's just 7 you're responsible for, right? Your, where'd it go? Pancreas. Does your pancreas just produce 2 hormones?
No.
Nope, produces 4, but you're only responsible for 2. All right, but I want you to notice something else. Look at the adrenal gland. Look at the medulla of the adrenal, or the testis. If you notice on these, you have basically 2 different lists here, right? So you've got that up here in parentheses, or you've got them here in the subsets. When you have this, what I am showing you is that there is a class of hormones, often of which there are dozens. And then here, subset would be one specific example. So testosterone is one specific example of an androgen. Estrogen is the class of hormones. Estradiol is the most common. Progestins is a class of hormones. Progesterone is the most common, right? If we roll back up here, glucocorticoids are a class of hormones. Cortisol is the most common specific one. Mineral corticoids, class. Aldosterone, specific. So on the exam, if I'm pointing at a particular region of the adrenal cortex, I may ask you for a specific class of hormones that is produced there, or I may ask you for a specific hormone that is listed there, right? Insulin is just insulin. Glucagon is just glucagon, right? So that isn't that challenging. But there are some that you need to know both the class and specific examples. So class and specific examples, right? Same thing here, same thing here, same thing here. So you've got class and specific examples. So there are a couple of those where you've got that class and specific examples. So be aware of that as well as you're going through that, okay? These aren't two different hormones. This is the class of hormones, and this is the most common one. All right? And because obviously that's not enough in and of itself, of course, if you go down, we have plenty of histology here as well. What I will say is that the nice thing about the histology that you have on this is most of the adrenal glands don't, most of the endocrine glands don't look like any of the other endocrine glands. Everything looks pretty distinctly different. So it is kind of nice that way. It isn't too challenging that way. So once you recognize them, which hopefully will be pretty easy, you'll be able to answer all the questions. So there is a lot of histology, more histology in this section than on the last one. A lot of histology. You haven't seen this much histology probably since the epithelial and connective tissues in 430. All right. So that is the game plan. That is what is expected of you. So this weekend, I want you to look at these processes that you all sat there quietly, glazed over eyes about for the immune response. Make sure you're comfortable and understand these processes, the activation, and everything that goes along with that. But it is also important to look ahead at the endocrine system as well. Because if you don't spend any time looking at the endocrine system, next week's going to be brutal. All right? Questions on any of that? All right, that is all I have for you today. We get to finish early today. That is all I wanted to talk about for today. Next week, both days are going to be like today, going to be long lectures. We're going to have a lot of material to cover, but we will get through it all to make sure that the following week, all you have to do on Tuesday the 6th is group presentations. So don't forget to work on those as well. All right. Any questions? All right. Have a safe weekend. Study hard, and I will see you next week.